Hello all. This is my first time using this particular bit of technology, Screencast Miner. So it may be a little rough, but I figured I would use it first time to get some practice in, to go over the last bit of Chapter 14 that we didn't get to in class. First of all, A, it may be a little rough. B, you may see me experiment with doing things like this, where I see if I can actually pause it while I'm talking, and then start it somewhere else, and then pause it again, and then start it in a completely different spot. I think I can. Okay, back to Chapter 14. So, how do we study this stuff? Mendelian genetics. Well, clearly, Mendel studied with pea plants, and he looked at lots of different characters, which had multiple different traits for each pea plant. And there are other ways that we study this too. We might use animal models like Drosophila melanogaster or rats. We're always used to hearing about lab rats, but why don't we use humans? Well, you could probably imagine our generation times are pretty long. So if you want to wait from one generation to the next, it takes you 20 or 30 years instead of a couple of weeks in the case of a fly. Also, instead of producing dozens or hundreds of offspring, humans produce relatively few. And then, you can imagine some ethical implications of doing breeding experiments with humans. So, how do we manage to study human genetics? Well, in some ways, human genetics are going to be similar to what we find in other organisms, so it's helpful to study them. But we can also do pedigrees, and these are family trees that we create that describe how parents and children across generations relate to one another, and they show patterns of inheritance. So this is an example right here of a pedigree, and if you look up here, hopefully you can see my arrow, you can see a first generation, and you can see a box and a circle. The square, the box, is going to be a male, the circle is going to be a female, and if it's shaded in, that means they are affected. Right? So this is two different couples in a first generation. Let's look at this couple here. You can see their genotypes, right, along with their phenotype, affected or unaffected, based on shaded or no. This particular disorder, if we look at the first generation and the second generation, we can tell that it's a dominant disorder, right? Because anyone, even if they have heterozygote traits, like big W, little w up here, they have the disorder. If they have little w, homozygous recessive, you can see they're not shaded in, they don't have the disorder. If you look over to the other side, we're looking at, instead of widow's peaks, earlobes, and for this trait, you can tell that the affected individuals all are homozygous recessive. And as long as you have at least one copy of that dominant gene, you are going to be unaffected. Right? In this case, affected, it means you don't have the trait. Right? I'm talking about these as if they're diseases, but they're not. Right? So, to the left, you have a dominant trait. To the right, you have a recessive trait. These pedigrees are useful if you want to follow this movement of a condition across generations through a big family tree. For instance, if you were to look at royal family trees in the 19th century in Europe, you would find a lot of recessive disorders that would pop up all over the place, and you could look at pedigree to see how they tended to be distributed and inherited, and you could tell by who had them and who didn't, whether they were recessive or dominant. And I've posted a video that you can see for this week's folder that will explain pedigrees a bit further to you, and if you have any questions, please let me know. So, I keep talking about widow's peaks and earlobes as if they're disorders. They're not. But that's because very often we talk about genetic disorders. And genetic disorders seem like they shouldn't exist after millions and millions of years of evolution, right? You would expect that if a trait is deleterious, if it harms you, especially if it's deadly, that over time it would become rarer and rarer in the population, and the healthy genes would spread and become more and more common in the population until eventually you wouldn't have that deleterious trait. Well, there are a couple of ways that you can have harmful genes that stick around in a population. First of all, mutation. You can have new ones that pop up by chance, and that does happen occasionally. But more often, you have recessive disorders. These are disorders that don't necessarily go away very quickly because you can be a carrier and not display any of the deleterious effects of that gene as long as you only have one copy of it, right? As long as you have one healthy copy, even if you have a copy that includes the disease-causing gene, then you're not affected at all, and you will have no problem creating offspring for the next generation. So if you are homozygous recessive, that means you are an individual that has the condition. If you are heterozygous, it means you're a carrier, right? So you carry the gene for the condition, but you do not actually have the condition. So what are some conditions like this? Albinism is one example. So you can see a Punnett square between two parents here. Both of them do not have the condition, right? Both of them are carriers, and you can see that about 1 out of 4 of the offspring will end up having the condition. And if you consider why a lot of cultures have taboos on marrying relatives, right? One reason for that is that incest tends to more often result in children with certain genetic disorders. Well, the reason for that is all of us have certain recessive traits that are probably carrying disorders. And so, if I carry certain disorders and my wife carries certain disorders, if we carry any of the same ones, then we have some likelihood of having a kid that has that disorder. And we just hope that we don't carry many of the same disorders. Well, if I were to have married my sister, since we have similar genes, she probably carries more of the same disorders that I do than my wife does. And then you're more likely to have a kid who's homozygous recessive for that particular disorder. So these are called consanguineous matings, and you tend to see, even in the animal kingdom, very little mating between close relatives. Now, it happens, but it's not too common. Now, albinism is not a big deal. You wear sunscreen and you're fine. But there are other recessive traits that can be a very big deal. So cystic fibrosis is a genetic disease that's caused by a single nucleotide that is mutated, and it messes up chloride pumps. And those chloride pumps are essential for keeping a watery layer in mucous membranes that don't get too sticky. And having sticky mucus layers means you can't move that mucus up and out of your body, and it can cause lots and lots of problems throughout your respiratory system and your digestive system. And about 1 out of every 2,500 people of European descent tends to pop up with cystic fibrosis, unfortunately. Sickle cell disease is another interesting example of a recessive disorder. But this one's a little bit different because to have sickle cell disease, you have to be homozygous recessive. So it is a recessive disorder. But individuals who are heterozygous have what's called sickle trait. So they do suffer some deleterious effects from having that recessive allele. But they don't have full-blown sickle cell anemia. And it turns out there's a side benefit. Individuals who are heterozygous are resistant to malaria. And so in areas of the world where malaria is common, so areas closer to the equator that are wetter, there's actually been some evolutionary advantage to having sickle trait, being heterozygous for this. And this was figured out by a scientist that was in Kenya, and he was a doctor, a practicing doctor, and he was comparing populations that lived way up in the plateau in Kenya versus in the lowlands. And in the plateau, it's far fewer mosquitoes, and therefore far less malaria. And it turned out sickle cell anemia was far less common up in the plateau, but when you move down to lowlands, where it's warmer and there are more mosquitoes, all of a sudden sickle cell anemia is more common. So that is an interesting evolutionary example. And so because of where malaria tends to be common, if you have ancestors who came from those areas, you probably are more likely to have genes for sickle cell anemia. And so African Americans are disproportionately affected by that disorder. Now, in some cases, there are disorders that are dominantly inherited. As you can imagine, this doesn't happen very often for lethal diseases, but in the case of achondroplasia, it's a type of dwarfism that is dominant. So if you're Game of Thrones fans, Peter Dinklage, I think, has achondroplasia, and all it takes is one allele to have that type of dwarfism. Now, this isn't that big of a deal in terms of reproduction, right? You can have dwarfism and you can have plenty of offspring just fine, and so it makes sense why that allele might stick around in the population. But what if you had a disorder that was lethal? Think to yourself, how could a lethal dominant gene stick around in the population despite clear evolutionary pressure to sort that gene out of the population? Well, in the case of Huntington's disease, which is one of the rare cases of a lethal dominant gene, it doesn't show up. Well, the gene is there, but it doesn't start displaying symptoms until after your reproductive years. So, Huntington's disease is a degenerative nerve disease that can cause you to be unable to move. You could start off with tremors and then eventually become unable to move, but it also can deteriorate your ability, your cognitive ability. It's a very, very sad disease. I don't know if any of you guys are into folk music, but Woody Guthrie had it. His mom had it. His mom set his sister on fire when she was beginning to come down with severe cognitive symptoms. And they didn't know what it was, but it turns out it was Huntington's, and they knew that because he got Huntington's when he was older. But because of that, it's able to stick around in the population because of the fact that it doesn't show up until after reproductive years. This is one example of a disorder that may have a chance to fix using gene editing technology that we'll talk about later in this course. Huntington's is a good example of a reason why having genetic testing can be nice, because if you have a parent that has Huntington's, you have, if they are heterozygous, you have a 50% chance of turning out to have Huntington's. Do you want to get that test, or do you want to not know? Well, it's nice to go see a genetic counselor to discuss those issues. And if you're a person who's interested in genetics and also interested in helping people, that would be a good line of work for you. Right? Okay, I'll mention one last thing before I finish this video, and it's fetal testing for genetic disorders. And there are two main ways that you can do this: amniocentesis and chorionic villus sampling. Amniocentesis takes a while before you're able to get results. It's less invasive, I should say. Chorionic villus sampling may actually go into the placenta and remove cells, so it's very invasive and a little bit riskier, but you get quicker responses and you can make a karyotype from there. Thanks, guys.